Hi guys, today we will discuss about the how to read the biological research paper by step by step. This video is based on that UG student, PG student, and beginner of the PhD students. That based on that how to do the research paper, how to analyze the research paper, how to read the research paper easily, and a step by step process. Okay, uh, it's not for uh, fifth year of the PhD student or last year of the PhD student and a postdoctoral student. It is only based on that UG, PG and uh, pre-PhD students also. Okay, we give the some we give the some guidelines to analyze the research papers using AI tool and basic information of the research papers. Okay, we will see them by one, slide by slide. We will discuss the outline of the research paper and how to separate the topics one by one and how to analyze the data one by one. We will do the separate. It is not like one video. It is more than one video, like the series of the process. Um, if you identify or analyze a paper very easily, quickly, analyze a concept, the theory and practical point of view. It will be you will get the easy tips and tricks also. Okay, we will see one by one slide. Okay, we move to the next slide. This is the topic heading. Okay. Hmm. Do you remember what is a research paper? Research paper means something they will be researching, they will be published the paper, right? That is correct only. It's not like a novel. Novel means it is based on the dream stories or based on the true stories, based on the like that. It will be that long long story like that is a novel. But here research paper means if you take on particular topic, you will analyze the topic, you will find out the new things in the topic, or you will find out the uh, new techniques or new tips or new factors. Anything you will find out the new things, that is called the research paper. Something different new, something will be different for that other research. That is a research paper. Okay, it is not like novel. Based on the research paper, what are the things will be they needed? Technical part and the information part, all those things they needed. Abstract is different, and research paper is different. Abstract means it will be short form of the research, outline of the research. That is called abstract. If you do that on research paper, that entire paper will be around 10 pages. You can summarize the paper at summarize the paper for to the 1 pages. That is called abstract. It is not complete research or complete exposure of the results. It will be outline of that process. Okay, that is called abstract. Basically, why I'm choosing this topic, I will explain easily. The example that every research scholar they doing research studies for 5 years minimum. They will be doing 5 years. In that 5 years, they will be have time also. They will be have money also. All those things they needed. But if they will be doing the initial stage of the studies, they will they become they become they struggled more things. With the 5 years, first 2 year or 3 years, they will be facing more struggles. Yes, because uh, they don't know if you completed, someone will be completed the masters in biotechnology or any life science student, they will be moving to that research field. In that research field, first 2 years they will be done for the core paper things. The second year they will be doing for the research paper. Some professors they are already doing for some research. They given for the some topic. They given assigned for the some topic. They modified the topic. They given for to that student. But they don't know if they will be studying the masters also. They will be studying the simple things, simple way, not much complex. Here the research paper will be complex one. You need to read more papers in the daily, but we don't have enough of time. We have to make the time. If example one student will be 1 week, they will be reading 10 paper, 10 different paper. That is a 10 means it will be very high. I give a maximum number because every paper they will be taking 1 or 2 days because they analyze the paper. They given the what are the title they given, how they will be given, where they will be find out the things, and what are they will be newly find out. This thing will be there mainly. They will be either for at least on paper 2 to 3 days. If you thorough that research, at least you need for 2 to 3 days. Uh, they needed. Okay. We don't have lack of time. If you using AI tool, it will be make it very simple, or you save more money. You have lot of time. You explore the more papers per day. If you using normally using 10 papers per week, example we take 10 paper per week. If you using AI tool, we get we learn or we read for that more papers per uh, week or month around like if you have 10 paper, it will be more doubling or tripling papers around 20 to 30 papers. You should identify read that time. Okay, that is the reason we use it for real tools. Okay, the read the line by line, that is a waste of the time. If you read the line by line and you have don't have mental energy and physical energy also. Okay, if good researcher will be there, example we take on paper, that paper will be using uh, cell culture. They given from some results they using for the some materials and methods, and they will be proposed some results. That factor also, the some factors they identify, the factors also it will be using for that studies. Okay, if that good researcher, they will be easy to access first that paper, and that paper they will be going to that materials and the methods and structure of the image or the graphs and their pathways, they will be find out. They will be getting easy idea for that presentation or paper, like that they will be identify easily. That is a big uh, theory. Okay, that is a good researcher they will be used. I will summarize it one more time. The good researchers, they will be going to directly going to results and materials and images pathway. They will be only preferred. They don't have lack of time. They don't have much time. They have a only lack of time. That's why they will be going directly going the research topics or research paper. Sorry, research images or research pathways like that. They will be going easily. Okay. Then we will move to the next slide. That is smart reading order. We have separated some reading orders. That is first for smart reading orders. That is initially if you read the one paper, they will be given for the some title and abstracts also. Title and abstract moreover similar. If you read properly in the title and abstract, you will be get to the paper around 30 to 40 percentage what they told, how they doing, what the research proposed. You identify easily. The title and abstract is very important For that, each and every paper you should read, you must know the title and abstract. Okay, what do you do? Okay, if the title means what they tell, what are that is a main topic. Title means there is a main topic. Like if you researched on antimicrobial studies, they given for which plant they will be taken. Example, they will be studied for the plant. Which plant they will be taken. What are the test identified? What are the given results? And which assay they performed. They given in the short form of that, the title and abstract. If you read properly the title and abstract, you will be getting around 30 to 40% of the knowledge that is needed for the title and abstract. Okay, and read the abstract carefully, because uh, what did they say, what did they studied, why, what did they find, it will you to access easily. Okay, that is a big that is a important one. If that I remember, I tell one more time also, title and abstract is very important. You should slowly read the title and abstract. You will get around 30 to 40 percentage of the knowledge in the research paper. Okay, okay, we move to the next slide. That is figure and figures. Sorry, figure and figure ligands. Figure and figure ligands. Example, if you take on newspaper first page, some newspaper they will be given more paragraph like that things. One paper they will be given same content, they will be given for the image like things or graph like things or comic animation like things. What do you remember, what do you remember that paper? If you take remember the first paper or second paper? Definitely we remember for the second paper. The animation image graph will be there. If you access and study it very easily in this part, that is they represent figure. Okay, it will be more approaches easily. Okay, here all the figures and the if you if you have on paper, okay, example, if you have on paper they given for the 5 images. Okay, that images like that pathway like thing, you will get easily to read and study it easily. Okay, because that papers are their research, they're studied, they're exposed, they're find out the new things. That is the research paper. They will be exploring that the research results in that graph format, table format, or image format. You will get easily to get the knowledge. That is a big reason. Okay, the figures are very important. First of all, the title. Next one, the figure. That is important. If you identify, if you read the figure, they given for the some caption or description. If you see in one image, example, if we take that on images, if you take some images, you should see for the image what they told, what they will be there, what are the things are there. Then read for the caption or description, what are they told, what are they find out. In this thing important, if you read on images, that description, you see the description and captions, you and then read side by side, you see the images also. And the some images they will be given for the methods and test resolution also. If you have easy to read for the paper, you will get the easily for the result, how they will be using, how the signaling pathways works like that. If the most of the signaling pathways like that in that flowchart or image like format only, the signaling pathways, if you read the caption properly or description properly, you will be get that images, or you will be get the knowledges very easily for that figures. That is the important things for the figure. You should must read for the figures also. Figures, it will be easily to memorize the paper and content and methods also. Okay, we will move to the next slide, that is introduction. We will go that first we will see for the title, next one we will see for first one we will see for the title and abstract. Second one we will see for the figure. Okay, third one for the introduction. Oh, introduction also it will be very important. Okay, one second, I will tell. The title you will identify the 25 to 30 or percentage of the topic, and other figure you will be find out the 70, sorry not 70, like 30 percentage, example 30 30. This one for the title will abstract for the 30, and the figure you will get the 30 percentage of the paper. 60 percentage you should know. Okay, next the introduction will be very important, because if you find or if you read the introductions, you will identify that around maximum the topic what they told, like that what they research, like that you will identify easily. That is a introduction is very, read the introduction very carefully, because why? Because you will identify easy the entire outline of the topic. What are they do, why they perform, why should read for this paper. You will identify easily. That is that introductions. Focus on the topic. What is a big problem? Sorry, once again. Focus on the topic. What is the big problem? They every introduction they mentioned that some problems they will be doing for this thing, like that. Example be there, someone studied for the Alzheimer disease. They given for introduction. Overall, that 1,000 people, around 10 people are affected from the Alzheimer disease. How they will be will be find sorry, around 1,000 people, 10 people are affected for the Alzheimer disease. We should prevent the disease. That is a problem. The Alzheimer disease will be spreading or not spreading. It will be the patient's ratio will be that increasing. How they will be prevented for the patient and a diagnosis treatment. That is a problem. Okay, that is a problem. Like that, like here also, every topic they will be given for some problems. What the problems you should identify, and already what is already know. They sometimes they people will mentioned already, they will be studied for that this thing. They will be identify this thing. We should study for the related things. That is they will be mentioned. That is also important. And what is unknown, some factors are unknown. If that is some disease will be there, if the some factors will be unknown, factors will be affected for that the disease, we don't know. We don't know anything, but some factors will be identified. They will be affected the factors. They don't know, they don't get the name or properly identification properly. We don't do anything, but some unknown factors will be there affected. We should identify that the unknown factors in their problem. That is important. And hypothesis and a goal. Okay, if the Alzheimer disease, example we take the Alzheimer disease, that is ultimate Alzheimer disease will be there. The problem will be there. The problem will be there means we should identify the solution in the research. Okay, that is that goal. If that Alzheimer disease will be, the patient ratios will be increasing, we should prevent or we should diagnosis, we should treat that patients, we should get that proper result for the research paper. That is a futuristic growth. That is that goal. What are they, why we will do, what are the process, that is a goal. Okay, that is important. That is mainly for they will be why the work will be there, the context format that tells you why did you studied, and I help your link and questions figure. Just that is not a matter, but these 4 points are mainly matter. One is for the what is the problem, what is already known, and what is unknown, and what is their hypothesis and the goal. These 4 points are very very important. If you read some paper, example, if you read some paper, you took read the properly the introduction for that 1 or 1 pages, you should identify that what are the problem already known, that already known, another unknown, the hypothesis, goal also you should identify it. Around example, anywhere you will take the any paper, you take that these 4 things are must should read. You will identify that around 80 percentage of the topic what they told identified. Okay, next we will move to the next slide. Some suggestion, what are things you should tell me. I will recreate, or I will give my best in this. This is my first presentation in the online. If you have any suggestion or anything, you can put the comment. I will read and reply. I will give shortly. If something you will change to me like that, just also you can put it in the comment. And it is something I will make myself better. Something make yourself better. Learning is a process. Okay, if you see this video, both things will be there. Learning is a process. Don't quit for the learning. The learning will be going. You will get more knowledge. What are things? If you read the biology, this you can go. If you read that uh, accountants or commerce like that, you can go. If you read there's some AI tools and developing, if you go, that is, if you should develop your skills, update your, okay. If you have some suggestion, if you have some queries, if you have anything, you can just put it comment. I will reply. I will reply. Okay. Okay, we'll move to that next slide. That is conclusion discussion. uh, Mostly the conclusion discussion will be uh, if that is important but not much. Because why I don't know. Some people's example, I will tell. If that one people will be there, this is A. One people will be there, another people will be there, B. Okay, they both are doing same research. Okay, both are doing same research, but they will be getting the results will be positive. One people, okay, another people on result will be getting negative. What they you should conclude? Think. If some people's getting positive result, some people's will be getting negative result. Both are doing same reagent, both are doing same thing, same steps, same process, same thing. All are those the same thing. Here also both are doing 5 years same work, biggest work. One people will be getting positive result, another people will be getting negative results. What they will be do? If A is correct, A is correct. B is correct, that is B is also correct. How will we find the that things? That is many factors are influenced by that in this condition. Okay, that is the main reasons. Okay, if you read that both papers, you will get more knowledges, same topic, but the results will be different. Okay, the research paper or article, all those things based on the somebody results. If that group A will be there, or sorry, group A means so A people will be there, and B, both will be doing same process, same thing, everything same, but they will be getting good positive result. They will, another people will getting negative result. Both paper also published, positive result paper also published, the negative result paper also published. What they told, they will be given for all those things same thing, but they will be getting proper results. Result not getting proper result, they will be getting this negative result. But here, no, don't take stress like that. This is simple thing I will tell you. If you read properly in the both paper, you will get maximum knowledge in the paper. Okay, what they told, what they conclude, you will identify easily. Okay, they will. I will give that. Skip the discussion and conclusions. If you read that title, figure, and introduction, you will get a maximized knowledge for this paper. Around 90% of the knowledge of the paper. Okay. The discussion and the conclusion based on the results. Some people is getting positive results, some people is getting negative results. Don't take that it will be good paper, don't take it will be bad paper like that. They will be expressed in the studies, they will be getting positive result, they will another people will be getting negative result. That is not an this is good paper, this is bad paper. You should read the both papers, you should identify both knowledges, how they doing, what they doing, you should learn everything. Okay. um, and the data is also important, data and the limitations, the future work, the mean what they finding, and the data and the interpretations, and the mainly field, and any limitations and open questions, and future works, that is important. They both will be doing for uh, same work, but they will be getting different results. Some people getting positive result, they people will be getting positive result, they will be expressed, they will be given for conclusion 1 point. And some b people getting same thing, they will be getting negative result, they will be concluded with some point. That is a research paper. Research paper means like what they doing some process, then they will be identify the things. That is a conclusion process. Okay. That is that main finding, and the data interpretation also really important. That is a future work also there. Okay. If you have confusion, anything, you can ask me. I will explain next video. I will tell you personally also. If you find this, we will move to the next slide. Hmm? Okay. Next, results. Result is very important. Now you go back to the result sections. Results is with you should looking for the figures, data introduction. That is, the results are very important. What they getting a result and what they think, so that is important. Okay. Next we will move to that methods. Methods are very important. Methods also important. Methods is, if you identify the methods, you will get the maximize knowledge. Around 100% will be done in this order, will be around 100% will be done. Methods, what are the techniques they will be using, what are the techniques which they should learn, and how they using the techniques, and little bit detail, it will be very important. That is a methods and the biology. Okay. And example, this is I take that one example paper. uh, you should see this paper example, and I will explain step by step how they will be doing, how they will be done by the process. I will discuss step by step. Okay. Here they image the images of the CRISPR editing in that zebrafish. This is a zebrafish doing for the CRISPR editing. This CRISPR Cas9. This is the most advanced studies. Okay, they will be doing CRISPR studies. Okay, title they given: efficient of the targeted gene knockout in the zebrafish using the CRISPR Cas9. Efficient of the target gene knockout in zebrafish, where they will be doing gene knockout process, where they will be doing in that zebrafishes. The zebrafishes they will be doing gene knockout. Used by that which technique? Used by that CRISPR Cas9 technique. Here they will be given which species or which plant, or like which animal they will be mentioned. What they will be doing? Gene knockout they will be doing. What they do in that gene knockout. Which method will be using? CRISPR Cas9 will be using. Okay, here zebrafish they will be taken. Sorry for the bad heading. One second. Here this they will be taking zebrafish. What they're doing? They will be doing gene knockout. Gene knockout. They will be doing. What they will method will be using for the CRISPR Cas9. Cas9 they will be sorry Cas9 they will be using. That is a process. If you identify the topic title, you will get around 30 percentage of the process and the paper. Okay, make it. You can write it to your note. That is 30 percentage of the topic you will get. And the abstract or the abstract scheme. Okay, they knock out gene studies in the eye development. Okay, abstract they will be measure in that gene knockout will be they will be doing. Where will be they doing? They will be doing in which place or which gene they will be mentioned in the gene knockout in eye. Where will be mentioned the gene knockout in eye development. Okay, it's clear. Okay, next we will move to that. Abstract is over. You will get around 20 percentage. You can write it. 20 percentage will be done. Okay, around you will get 50 easily, very easily. And the figure, look the image, see the photos in the embryo and graphs mutation efficiency. Here they we don't mention any papers anything, but we will take only title. If you see here images, if you see in the photo embryo, if that embryo will be there, this that sorry, the zebrafish embryo will be there. They mention the mutation efficiency studies in that graph, how they will be doing, how they will be acting, how they will be pathway will be interrupted, what are the mutation will be happen in that happen. They mentioned by the graph or images like that. You should look out the images easily and identify that mutation studies. Okay, that is you will get around 25 percentage of the topic. Okay, now we will get 75 percentage of the topic. Okay, now that introduction also we should very important. Read that unknown genes. Read what the unknown genes will be there, why they do want that knockout. If that if there's some more genes are there in that zebrafish, but we should do that. What you know that the genes and the knockout. They want to knock out the genes like the process. Okay, that is very important. Introduction. You should read the introductions. You will get the what are the genes and where will be knockout, and that is important. They will be mentioned that introduction part. You will get around 20 percentage of that complete topic in this introduction. You should read the properly in the introduction. You will get 20 percentages. Around you will be get 50, 75 and 20, 70, 90. Yes, 95 percentage you will get. Okay, next conclusion results method. You will be get around what the percentage, 5 percentage you will be get. Okay, here conclusion means read the conclusion, what they find, what the genes affected the formation expanded, what they given the conclusion. If you get if you they, what are they get that result, will be given for the conclusions. Okay, that is a conclusion. And the scan the with the measure the gene editing and evidences support the climbing like that. And optimization, what the techniques will be used, they will be using the guide RNA. Here you will be using guide RNA and Cas9 enzymes, CRISPR method, and how they will be injected to the zebrafish, how they acting. That is that methods and materials and method. That is the injection method. How the injection, that is some many methods are there. The inducing the induction process, they will be induced in that geographies. And what are the mRNAs will be used? Some kind mRNA they are used in the CRISPR-Cas9 process. They will be mentioned here. Okay, you should see here the title, and you will get 30 percentage. Abstract you will be getting 20 percentage. 50 percentage is over, and another 50 you will be get the figures 25 percentage, introduction 20 percentage, and the result methods and conclusion for 5 percentage. Total 50 100 percentage you will be get for the this paper. What is not this paper, you will get any paper you will take, you will get this order of the topic. You will be get for that, uh, surely you will get the 100 percentage knowledge in this paper. If you don't do that, if you don't do that, without order, sorry, if you don't do that without order, following the order of the topic, you will be get the knowledge. That is the true. If you align the topic title, abstract, figure, introduction, conclusion, results, method, you will be get that more knowledges. Okay. Okay, it's clear. We will move to the next slide. The summary. We will come to the last slide. Summary. Okay. What are they? We will study. We should conclude all those things one more time. Here, first title is important. Abstract is second important. Figure also important. Introduction important. Conclusion, result, and methods also important. You should read properly, for in this way you will get definitely 100% knowledge in this paper, and you will get uh, more easily to learn further complete research paper easily. Next upcoming videos we should discuss for that AI tool in that research paper. If you using AI tool, that is the main topic. Next video is for the main topic. If you using AI tool, you will be getting 1,000 time, not 1,000, it's more than 1,000 time. You will get more knowledges, more time saving, and more efficiency of the research paper. Okay, this is outline of that research paper. This is outline of the research paper, and we will this is not a one video, a series of the video. You should read the playlist completely. I will make the playlist, and you should click and read the completely biological research step by step. You should learn for more things, and you should easily to access all the paper easily. Okay, that is things. Okay, if you have any suggestion, if you have any comments, you can give the comment that inbox, that also that comment that video description also. I will reply. And if you have any queries, if you have anything suggestion for any topics, or if you have anything changed, or if you have anything want to make, you will tell that comment openly. I will reply. If you have anything, you can contact me. Okay, bye. That is the first video. We will discuss step by step in next videos. Okay, thank you.